Alright, so here we have a DNA replication diagram with all of our terms already written out. We're going to go through and label where they belong on the actual diagram. So the origin of replication is this whole thing. It's where the DNA replication is occurring on the DNA strands. So we have our 5' end and our 3' end. Since the DNA double helix is anti-parallel and complementary, that means this is going to be our 3' end and this is going to be our 5' end. And when the replication is occurring on either strand, we can have our 3', 5', 5', 3'. Okay. And then we have our leading strand, which is this strand right here. Our lagging strand, which is this strand right here. Leading strand is going from 5' to 3'. That means that has to be our lagging strand. And our guy race, that's going to be located right here. And that relieves the tension and prepares for the breaking of the bonds in the double helix. And for helicase, which is right at the replication fork, and that breaks those hydrogen bonds and leads into the single-stranded DNA. Then we have our single-strand binding proteins, which are on the ends of these pieces of DNA that help to keep it apart and allow for replication to continue and to occur. And then we have our RNA primase, which is here on our 5' end of our lagging strand. And we have our Okazaki fragment, which is like this whole fragment, this whole fragment. It's a discontinuous replication on the lagging strand. And then we have our DNA polymerase 3, which is on this side on the lagging strand, and on the 3' on here, also on our leading strand. And then we have our DNA polymerase 1, which is found around here on our lagging strand. It places the RNA primers with the DNA nucleotides and correct any errors and fills in the gaps in the fragments. And then we have our ligase, which comes behind our polymerase 1 and cleans up any nicks and gaps in the DNA to help make it one continuous strand. And then lastly, we have our phosphodiester bonds, which are here and here. It's between the 3' and 5' ends. It creates one full bond and complete hydrogen bond to complete the DNA double helix. And the ligase is found like down here.